Hello, everyone, and welcome to T1D to 100's series on what's going on in research. Thank you all for being here this evening, and I'm pretty excited. This is kind of hot off the press news and hot topic and things that are just important to our lives. So, welcome. And tonight we're talking about new cell-based therapies for T1D edging towards the cure. Do we say that? I don't know. Um, before we begin, medical disclaimer, this presentation is for informational and education purposes only. It is not intended as medical advice. Please consult your healthcare provider before making any changes to your diabetes management and treatment. Not that this presentation is going to change your diabetes management, but we just always say that. Rules for this discussion this evening is your questions are welcome. We already have several that are queued up. Please put them in the chat, and we have some of our admins who are here that will share the questions with our presenter. As best as you can, be respectful and supportive of everyone's experiences and questions. And this session is being recorded and will be shared on our YouTube channel. You can see it on the screen, and it should be up in a day or so. So let me move along and just talk a little bit real quickly about T1D to 100. We are a community all about supporting, educating, and empowering people with T1D. We believe that living well with this disease at every age is possible and that no one should have to navigate it alone. That's why we're here. We are in a community where more and more people over 50 are getting T1D or living with T1D. We're the largest demographic at this point, and it's growing. But the resources and support haven't kept up with it. And aging with T1D also brings some very unique challenges that deserve attention from us and the medical community and the researchers. Knowledge research community, we connect people who truly understand what it means to live for a long time with this disease. We're supporting and involved with cutting edge research and some of the expert voices in our T1D community. Really happy that we can fill that slot and be available for whatever anyone needs. I want to mention on the team tonight, we have Kelly DeFerme. Did I pronounce that right? She is working with me as we organize T1D to 100 and get it ready for all the things we're beginning to plan for this community. And she lives with T1D and has just been an outstanding help. And she's online. So is Jim Cheers, who's been with me from almost the beginning and has been an outstanding support in growing our website. He is also a T1D. He's a bike rider. He's also an RN, and he's also a data scientist, and he's just a really, really nice human being from Washington State. And Jamie Lukov, who is also a T1D and um, works in the research community as an epidemiologist. And it's exciting. We have just a lot going on here and great people to interact with. So let me introduce our speaker tonight, Dr. Leonardo Ferreira. He told me how to pronounce it, and it was exactly what I was doing. Is assistant professor of microbiology and immunology at the Medical University of South Carolina and the Hollings Cancer Center. He earned his PhD in biochemistry from Harvard and completed a postdoctoral fellowship at UCSF, that's San Francisco. His research focuses on understanding how the immune system distinguishes itself from non-self, interesting, and to develop engineered immune cell therapies for autoimmune diseases, cancer, and aging. His work has direct implications for the future of type 1 diabetes treatment. This is really exciting. Thank you, Dr. Ferrer, for being here this evening. I'm going to stop sharing and turn this over to you. And thank you, and everybody, just take this all in. This is really interesting information.
Well, thank you for having me. I'll go ahead and share my screen. So you let me know if you can see. I have some slides that can be helpful. So it is a yes, you can see it.
Absolutely, 100%.
Perfect. So this is a picture of Charleston, South Carolina. This is where I live currently. Um, so today we'll be talking about new potential paths towards a cure for type 1 diabetes. And so we like to think of the immune system as a protector, right? It defends us from viruses, from bacteria, from cancer cells. As that slide earlier said, this self, non-self balance, it really helps us have an identity, right? What is self is to be protected. What is non-self is to be destroyed. And so you'll see a lot of pictures like this out there where it's just, putting a stop to invaders. But unfortunately, nothing is perfect. The immune system is no exception. So sometimes the immune system goes rogue and attacks self, attacks the own tissues. And so this is what we very broadly call autoimmune disease. And in a way, autoimmune disease is the body at war with itself. There are almost 100 different autoimmune diseases. You have rheumatoid arthritis, multiple sclerosis, celiac disease, pemphigus vulgaris, and of course, type 1 diabetes. So, in type 1 diabetes in particular, what happens is that the insulin producing beta cells in the pancreas get specifically recognized and killed one by one in type 1 diabetes patients. If you don't have this hormone insulin, you can no longer control your blood sugar. And so now you depend on this daily routine of pricking your finger, checking your blood sugar level, calculating how much insulin you should take, calculate how much you eat, and then repeat. And now, of course, technology has come a long way with continuous glucose monitors, but the risk is still there, right? If it runs out of these monitors or something else happens, you still need that exogenous source, that outside source of insulin. So again, I don't need to tell this room, right, that as a child, it can be pretty devastating because now, even before you know how to read or write, you are very aware of mortality, for lack of a better term. Or like if you do something wrong, something really bad could happen. And really your schedule and then also your parents' schedule revolves around a lot of these things. And so again, it's a pretty important disease. And so we would like to know how can we stop it from happening, right? So how can we go about this? So since quite a few years ago, I would say the 90s really, we've known that, if the problem is lack of insulin-producing beta cells, well, just put more in. And so working in the 90s, there's a paper here from the year 2000, the turn of the millennium, showing that if you take a pancreas and you digest it in a Ricordi chamber, and you take the islets, which contain the beta cells in them, and you inject them in the liver via the portal vein. Now, these new beta cells, they actually engraft. They make their new home in the liver of the patient, and they sense blood sugar just fine, and they make insulin just fine. And so this is already a functional cure for type 1 diabetes. Now, of course, there's a few issues with this. One is that there is really a tremendous shortage of beta cells for everyone. If you think about a heart transplant, it's a one-to-one, right? One organ donor, one organ recipient. If you think about a kidney transplant, it's a 1-to-2, right? One kidney donor can give two kidneys, so one to each person. But in pancreatic islets in beta cells, sometimes the other way around is like 3 or 4 to 1. You need to combine the islets of four different cadaveric, donors to be able to have enough cells for one recipient. On top of that, the disease started exactly because the immune system killed the beta cells. So if you just put more beta cells in, it's a bit like building a beautiful sand castle right by the ocean and just watch the ocean waves wash it away. Right? The problem did not get solved just because you put more beta cells in. Now, of course, there are some solutions to this, some of them many decades old, and these are the so-called immunosuppressive drugs. These are textbook molecules at this point that they really shut down the immune system as a whole. You've heard of steroids, tacrolimus, cyclosporin, rapamycin, and so on and so forth. And so these small molecules, what they do is they shut down the immune system as a whole, mostly T cells, T lymphocytes. And so on one hand, this is good because now the transplanted beta cells, or for that matter, could be a transplanted kidney or a lung or a liver, does not get rejected. On the other hand, it becomes severely immunocompromised. It's almost as if you have AIDS. You're very susceptible to bacterial infections, viral infections, to blood cancers. And so, of course, again, if you're a liver transplant recipient, it's either this or dying, right? You really don't have much of a choice. But for type 1 diabetes patients, technology has come to a point where you can manage the disease with insulin injections, diet modifications, really constant surveillance, and you might then be able to do without organ transplant without having to go through this. And this is especially important in the case of children. So, of course, these drugs come with lots of side effects. Some of them are across all ages, but some are especially nefarious for children, right? Imagine if you start taking these drugs when you're 5, 6, 6, 7 years old. There have been documented delays in growth, intellectual development, and so on and so forth. And so, again, we would like to not have to use this blunt, very all-encompassing immunosuppressive drugs to really bring this idea of beta cell replacement to as many people or every type 1 diabetes patient as much as possible. So there has to be a better way of doing things, right? So, and there does seem to be the case indeed. So we know from nature that it is possible to inhibit not the whole immune system all at once, but instead specific parts of it. And so that's what we want, right? We want to protect the beta cells without hampering immune responses against anything else. So we know this from many decades ago. Peter Medawar, Nobel laureate in 1960, he must have lived in a farm, I reckon, up in Britain. And so he did these beautiful transplant experiments in rats, in roosters, you name it. And so what he showed is that if you have a pregnant rat, or if you have a chicken egg, and you expose the embryo, right? So not born yet animal, to cells from a different individual. Now the embryo will be born and grow up and be tolerant, accepting of the cells from the individual whose cells he was exposed to before being born. So there's some critical time window where the immune system is learning still what is self, what is non-self. And so if we are to access that window, we can really re-educate the immune system. And so in our case, we would like to re-educate the immune system of a type 1 diabetes patient to no longer attack beta cells. Now, of course, we can't really go back in time and inject ourselves with stuff before we were born. But luckily, this phenomenon has since really been further dissected. And so we're no longer in the 60s. So now fast forward a few decades into the 90s. And what was found in 1995 more precisely is that within all T cells in your immune system, most of them do what you think they do. They kill cancer cells, they kill virus infected cells, and so on. But there's a subset of T cells that they're actually more involved in the business of peacekeeping, of keeping everything in check. And those are the regulatory T cells, or Tregs for short. And so this was work by Sakaguchi in Japan. And what he found was that if you take out this population, this group of cells from a newborn mouse, the mouse gets very sick with autoimmune disease. The mouse gets type 1 diabetes, it gets gastritis, and several other disorders. But if you now instead put these cells back in, now the mouse is happy. So something about these cells is pretty much preventing autoimmune disease from taking place. So maybe if we use these cells, since they seem to be responsible for the phenomenon of educating the immune system, maybe then we can have a living drug, a living therapeutic for type 1 diabetes and to really, protect organs and tissues that would be otherwise rejected by an immune system. So those cells, the regulatory T cells, we now know that they come from the thymus, and the thymus is where all T cells go to school. And so the regulatory T cell is no special in that regard. It goes to the same exact institution that all other T cells do. However, unlike most T cells, the regulatory T cells or Tregs, they instead express a specific set of genes, FOXP3, CTLA4, C25, that allow them to now be suppressive, to suppress immune responses instead of mounting immune responses. So those are the regulatory T cells that we'd like to work with. And don't worry about all these names, it's just to impress on you that these cells inhibit specific immune responses using well over a dozen different mechanisms. And so if we are to really take this into fruition, these cells are extremely unlikely to ever be replaced by a single small molecule or antibody. You need the cell. You need the whole cell to be programmed to go to the right place, to use different mechanisms depending on the situation, right? In some contexts, as you might imagine, some tricks might be more advantageous than others. And so you need this living drug to really take care of business in different contexts. And so that's the regulatory T cell as potentially a living drug for type 1 diabetes. Now, let's just do this then. So this was done, of course. So you can take type 1 diabetes patients' blood, you can purify regulatory T cells. We have surface markers on the surface of the cell. It's a bit like going to a supermarket with a QR code and you scan. Likewise, you can also pick in the blood what cells you want to put in your tube. So you get those regulatory T cells, you expand them into as many as possible, and then you inject them back in. And so the idea would be that, if in type 1 diabetes there are not enough Tregs or they're not quite functional, let's just take them out, grow them a lot, and put them back in big numbers. Some people have received up to 3 billion Tregs, and that's 3 times 10 to the ninth cells in a single patient, a single person. So the idea is there. Now, unfortunately, it didn't quite work. In this phase 2 trial, 110 patients, the results got published almost 2 years ago now. You see that there's no difference whatsoever in the amount of C-peptide and hence insulin, irrespective of the patients being a placebo, low dose, or high dose regulatory T cell injection. So I thought that if you inject Tregs, you're going to re-educate the immune system, and now it's going to stop attacking the beta cells and it'll all be good again. Well, that's not what was found. And many of us think it's because if you just take Tregs from blood and expand them, the likelihood, the chance of some of those cells actually recognizing, actually being specific for beta cell antigens is very low. It's like 1 in 100 million. And so, sure, you inject patients with 3 billion Tregs, of which maybe 30,000 are actually the ones you care about. Everybody else is just taking up space. And so we need to have antigen-specific, laser-focused Tregs to really help protecting beta cells from immune destruction. So how can we do this? Well, if they're so rare, it's going to be very hard to find them. So, okay, so then maybe we can find a way to create specificity in Tregs. And so that is a domain of what some of us call synthetic immunology, right? We're not restricted to genes that exist in nature. We can splice DNA together and make our own genes. In this case, one that we like making is called a chimeric antigen receptor, or CAR for short. You might have heard of CAR in the context of cancer, CAR T cell therapy. It's been approved since 2017 by the FDA, and it's cured many people from blood leukemias and lymphomas. And so, can we take this same technology now not to kill cancer cells, but to protect beta cells instead? And so, this is what the molecule looks like for those of you who are curious. So, a normal endogenous T cell, if you will, it takes two sets of molecules to be fully stimulated. You have the T cell receptor or TCR. That's what really binds these pieces of proteins. And that's really how the T cell really distinguishes self from non-self. You kind of have an ID card right there in the major histocompatibility complex, MHC. And then there's also some encouragement, and that's what we call co-stimulation, which is there with CD28. So that's a regular T cell needs these two things going on to really become, with energy to work. Now, if we have this artificial molecule, this chimeric antigen receptor or CAR, now everything is a one-stop shop. You have the recognition domain right there outside the cell. This can stick to any molecule I want. I want it to stick to a beta cell surface molecule, for example. And then this is the intracellular domain, also known as signaling. So this is how cells make decisions, right? So when this cell expresses this molecule, every time this domain binds to the target, boom, that sends a signal. And the regulatory T cell becomes stimulated, becomes active, becomes protective. So that's the molecular detail of how we can target cells to anywhere you want in your body. So we have this, handshake, if you will, of the CAR and the antigen on the surface of the target cell. So in our case, of course, we want to focus on the beta cells. So in type 1 diabetes, once again, you have your autoreactive T cells destroying the beta cells in the pancreas, so it can no longer produce insulin, blood sugar goes up. Now you want to send these tiny firefighters, laser focused CAR Tregs, so regulatory T cells engineered genetically to have a chimeric antigen receptor on their surface that can then make them go to the beta cell. And so now these tiny firefighters will inhibit these autoreactive T cells from destroying beta cells. And so if this is the case, you now restore beta cell function and you can now make your own insulin and control blood sugar. Now, of course, depending on at what stage the disease is diagnosed or treated, there might or might not be beta cells left to protect. And so this is why to really have a cure or a treatment for type 1 diabetes that works for everyone, it has to be a two-pronged approach. You got to address the beta cell replacement part of the equation, right? There's beta cells missing. You got to restore beta cell mass and also the immune part of the equation. You can't just put more beta cells in, as we said already, and just hope for the best. You also have to inhibit the immune responses that will cause destruction of those precious beta cells. So we could then do that. So we can have a CAR Treg, an engineered regulatory T cell that will protect a beta cell from immune attack from different immune cells. Now, it is very hard, if not impossible, to find a gene that is only present in one cell type in your body and nowhere else. Evolution is very thrifty, and so it likes using the same genes in multiple places. Like why invent a new gene if I already have a perfectly fine gene right here? And so because of that, it's really hard to next to impossible to target a unique organ, a unique type of cell in your body without worrying about any distractions, any off targets. And so we thought, well, the same way that we artificially conferred a new specificity to the regulatory T cell, maybe we can likewise confer a unique flag, a unique bait on the beta cell to be recognized by my engineered regulatory T cell. And so that's really the key concept, is that you are co-engineering a protective, a bodyguard, if you will, regulatory T cell with a beta cell to be protected. So that's really the core concept here. Now, this unique target, as I said, we're just going to make our own. And so we went to the whiteboard or blackboard and thought, okay, so what should this bait on the beta cell look like? Because we get to decide. We can't just design this DNA in our computer. So what should it be? It should be on the surface of the beta cell. Otherwise, the Treg can see it. It should be biologically inert, meaning that it should not hurt the beta cell. Beta cells are very stressed out cells already as is because they're constantly making insulin, storing and making some more and secreting it. And so we don't want to mess with a delicate beta cell too much. Should also be immunologically inert, right? The last thing you need is to create an immune response against the molecule that you're putting in the beta cell. That really defeats the purpose. And so what this means is that it cannot be a molecule taken from a jellyfish or a rat, for example, right? It has to be human or human-like molecule. And then last but most obvious, it should be unique. It should not be expressed in any other cell type in the body. So after quite some thinking, we came up with this truncated, this chopped off, if you will, epidermal growth factor receptor. So EGFR-T is biologically inert, immunologically inert, and unique. And this is very important because, again, the idea is that if EGFR, the full length, the total protein, is not immunogenic, neither is a piece of it. And moreover, as you see there, there is no intracellular domain in this protein down there. That's the lipid membrane on the cell. This is the inside of the cell. There's nothing there. So this protein is biologically inert. The beta cell does not even know that it's there. So once we have that, we can then have this bait, if you will, the EGFR-T, and now we come with a receptor for it, which is a EGFR specific CAR that we can put in regulatory T cells, which express FOXP3. And so this is what we want to test. We want to test if this arranged marriage really works, right? Do these engineered CAR Tregs talk to the engineered beta cells? Now, one little detail I didn't mention is that I keep talking about beta cells, engineering them, but I started out by saying that beta cells are very rare. There's not enough beta cells to go around. So if you're going to start messing with your genes, there will be even less. Well, luckily the field has now been able to make beta cells from scratch in the laboratory from stem cells. We can make enough beta cells to fill up a whole room. So beta cell shortage should no longer be a thing. These beta cells are almost as good as if they were to come from a person. And so this is really where we collaborated with Holger Russ's lab at the University of Florida. They are stem cell experts, genetic engineering experts, and so they really perfected the production of insulin-producing beta cells from human pluripotent stem cells. And so you see these skittles, if you will. This is in the laboratory. You convince the stem cells to go stage by stage, step by step into beta cells. And they know that their cells work really well because they have a green fluorescent reporter in their beta cell, such that every time insulin is made, these cells light up green. And so there they are. No shortage of insulin made by these stem cell made beta cells. And of course, the insulin production is not always on, always off. Depending on the level of sugar, it changes. So if you have low sugar, LG, insulin production is low. If you have high sugar, high glucose, HG, it peaks every time. And so these are functional stem cell derived insulin producing beta cells. Now, having this starting from stem cells, we can then really change any gene we want in the stem cells. And so we chose, as I mentioned, to introduce this one gene, EGFRT, to be on the surface of the cell. We also added another gene called luciferase that I abbreviated by Luke there. And so luciferase is taken from fireflies. So this allows the cells to glow in the dark. And this will be useful later on because when I do experiments in a laboratory, I want to keep track, to keep an eye on these beta cells without having to cut the animals open to look at them. So this allows us to track them without making incisions in the animals during time. And so that's the gene edit in the genome. And then we want to make sure that this is fine. So if you look at these pictures, what do you see between top and bottom? They look the same. So the stem cells do not even know that they have this new molecule. They look just the same. The stem cells and the beta cells, the glucose responsiveness is the same. You see the same exact peaks of insulin in different conditions. And so these engineered stem cell-derived beta cells are indistinguishable from the original wild type unmodified cells. So this is great. We have very nice functional stem cell-derived beta cells with a bait on their surface. Now, on the immune side, we take patients' blood, we purify the regulatory T cells, and we modify them now with a CAR. And so this is an anti-EGFR CAR to bind to the EGFR T. And so that's modification efficiency there. We have a red protein called mCherry in there. And so you see very nicely, you have almost 90% of the cells have your gene of interest. And then importantly, they remain regulatory T cells, right? By doing this trick, we're not changing the nature of the cells. They still express very high levels of that FOXP3 gene that I told you about. And here is a bar chart to help with that. Now, of course, that we have the characters, if you will. How do we make sure that they talk to each other? Well, there are molecules that go up, they appear in Tregs once they become exposed to their favorite antigen. In this case, one of them is called CD69. And so what you see there is that anytime we have the CAR Treg with the stem cell-derived beta cell, there it goes, it goes up. This means that they kissed. This CAR Treg saw the stem cell-derived beta cell with the bait, and then this went up. Moreover, this also made the CAR Tregs secrete anti-inflammatory cytokines. One of them is IL-10, interleukin-10. This is one way by which Tregs basically put some immune cells to sleep to make sure they're not causing any more damage. And so these little bars here demonstrate that the CAR Tregs are talking to the EGFRT expressing beta cells. Okay, now this is all good and dandy, but it's in the petri dish, and we're not made of plastic. We're made of cells and tissue and bone and flesh. And so you always want to have a proof of concept for these new ideas in an animal model, in a living organism. And here's where the humanized mouse model comes into play. You have these mice that we call them NSG for short. These mice do not have their own immune system, really. They don't have their own T cells that can make antibodies. That means that we can transplant into these mice any human cell we want. And so what human cells we want? We want these mice to have human beta cells, human islets, and human T cells. Conventional T cells to simulate the destruction of the islets, and engineered regulatory T cells to hopefully protect those islets from the other reactive, the bad guys, the T cells. And so this is the humanized mouse model of type 1 diabetes. And so this is what the mouse comes looking like when you put all these human cells in it. So what does the data actually look like? So the system is, because mice are small, instead of putting human beta cells in their liver, I put them under the kidney capsule. Equally highly vascularized, means lots of blood supply, and makes it easy to find, right? If I go later on, I can easily locate those cells right on the top of the mouse kidney. So you have a mouse, you destroy its endogenous, its mouse beta cells, so the mouse becomes diabetic, but then you put in human beta cells, and luckily for us, human insulin acts on mouse tissues. And so now this mouse can control blood sugar thanks to human insulin made by human beta cells inside this mouse. We let that heal, and then, just like in a patient, we're now going to have an immune challenge. And so here, for simplicity, we have two groups. One group of mice with the human beta cells is going to get human T cells that are specific for the beta cells. This means that we expect that in these mice, their beta cells will be killed, destroyed. Nothing left to tell the story. In another group, we have this, autoreactive T cells, but also some hopefully protective CAR Tregs. And here the question is, do these CAR Tregs protect beta cells from T cell destruction? Because if they are to work in a type 1 diabetes patient, they better be able to protect beta cells in a much simpler organism, much smaller too. And so that's really what we're testing here. In an in vivo organismic model, can these CAR Tregs protect beta cells from immune rejection? And the answer is yes. If you look at the glow-in-the-dark trick I mentioned, the luciferase, you see over time, if you have just these effector T cells, the killer T cells alone, if you look roughly 3.5 weeks, almost 4 weeks, there's very little to no beta cells left, right? Almost no signal in these mice, in this little heat map. But if you give the Tregs, now you still see the mice still have the signal there. And this is 3 mice as an example, but of course we did more than that, and that's the curve there. And so you see the black curve is the mice that receives only the killer T cells, and you see the beta cells get destroyed, no luciferase left. But if you get CAR Tregs, then you see nice protection. You see like an almost flat curve. And so this is what this data suggests, is that you have a CAR Treg that is locally protecting the beta cell from immune attack by these T cells, by making all these anti-inflammatory cytokines molecules that Tregs do. And so this shows that. Now, if you're a septic like myself, you think, okay, you're showing that you protected something inside the mice. But how do you really know that those are still beta cells, right? Maybe these cells are alive, but they scarred beyond recognition. Maybe they no longer make insulin, in which case they are useless to us in our quest for this new treatment. And so seeing is believing. We cut into these grafts and do antibody stainings by immunofluorescence. This is what we see. If you look at the mice that got treated with only the killer effector T cells, you see very few to no insulin producing beta cells. You see a lot of T cells with a C3 marker, and you see really almost no regulatory T cells. This is a deeply inflamed, deeply destroyed beta cell graft. But if you look at the mice that received the Tregs, now you see these very beautiful insulin-producing human beta cell clusters. And yes, you still see some T cells, but much fewer, and a lot of them are actually the good kind, the regulatory T cells, based on this purple FOXP3 marker. And so you really were able to rewire, change the environment of that beta cell clusters to now being protected from immune rejection. And so with this is a quantification. If you don't believe me, just from one photo, if you look across different mice, different fields of view, you see, you go from 0 to 50% of the T cells being regulatory T cells. The EGFR magic marker is still there protected, and the total number of T cells itself, as you saw by the red marker, goes down. And so again, it's a very different environment when you put the protective CAR Tregs in versus if you don't. So now that we have this, can we make new ligands? And so it's just a little side note, but just to tell you that we didn't really stop there, right? There's different locks and keys, if you will. If this is a lock and a key system, we might have to do different locks and keys to really work in different patients or make it even better. And so we kind of on this quest to make ever more precise interactions between the beta cell and the regulatory T cell. And so at this stage, you really only have in vitro data, so in the Petri dish, that T cells get activated or they stimulated by the right thing. And they also have this suppressive inhibitory function. The less peaks you see, the less the effector T cells divided, which is a good thing, right? You don't want them to make more of themselves to not cause destruction. And so we've shown that, but really this is what we have, right? So we're doing these experiments now where we're trying to be more and more sophisticated, right? So this is really getting into the weeds, but instead of having the mouse that I just showed you, you can make an even more human mouse. You can have a mouse that has multiple human genes in it, and then is also missing some mouse genes. And so this will be work with Dr. Mike Bram at UMass. And so the idea here is again is to have an even better model of human type 1 diabetes in the mouse. And so in this case, the idea is can we really protect these stem cell-derived beta cells from immune attack in this better humanized mouse model. And so using some of this what we call protein language models. So like you have these large language models like ChatGPT, you can imagine proteins made of amino acids. You can use those characters to also design new binders to go there. And so that's really been really exciting work that we've been doing with mostly the Shaturji lab at UPenn, University of Pennsylvania. And so this is where we have it. So this is where the concept is we have a beta cell, and to really allow it to be protected, you have this bait on its surface, which then serves to be recognized by a protective Treg, which allows a local immune protection of this beta cell. So that's the concept there. And so then the vision is to have this combination cell therapy for type 1 diabetes. You can have an off-the-shelf stem cell-derived beta cell, right? Those stem cells we use, they can be right off the shelf. But then you need to have a patient-derived Treg that gets engineered to protect those off-the-shelf beta cells. Now, of course, this is just the beginning. We could also try to make the whole thing off the shelf, which will make it more scalable for more people. And so there's now groups in Japan and other places that you can also make stem cells into regulatory T cells. So instead of making only beta cells from stem cells, you can also make the CAR Tregs from stem cells. And so now this promises to be a fully off-the-shelf system where you can have ready-to-go beta cells and ready-to-go regulatory T cells to protect those beta cells. And so that's really the ultimate vision for this approach. And that then the idea then is that these lock and key system will locally protect the beta cells from immune attack. And so we have this concept. Now, of course, you can put all your eggs in one basket, and there's also other approaches out there. And one of them that deserves mention is the idea that you can make the beta cells themselves also take matters in their hands and do what people like to call hypoimmunogenic. And so in a previous life, before I worked on regulatory T cells, I worked precisely on making stem cells and their derivatives, such as beta cells, into cells that are more invisible, if you will, to the immune system. And so now you are not only engineering regulatory T cells to re-educate, to protect the beta cells, you also change the beta cells themselves to have some combination of genes in and out to make them potentially more resistant to the immune attack on their own. And so this will be highly synergistic approaches. And so the most advanced work on this is with a company called Sana Biotechnology, which is based in Boston and South San Francisco and Seattle. And so the idea there is that, again, we've learned so much immunology over the past 100 years now that we pretty much know where every immune cell type binds to, right? So if you want to prevent a T cell from killing you, you're going to eliminate MHC expression, the major histocompatibility complex. And then is a detail, but there's two flavors of T cells, CD4 T helper cells, CD8 killer T cells. And so you can knock out not one, but two genes to really make sure that these cells are really invisible to T cells. That's B2M and C2TA. You also have to worry about macrophages. Macrophages eat other cells. So if the macrophage even has a suspicion that there's something wrong with you, it's going to eat you. And so if you have a foreign beta cell, a stem cell-derived beta cell, it could get eaten by the macrophage. So once again, gene editing. We can introduce CD47 that will suppress. It's a don't eat me signal to prevent macrophages from eating that cell. And also, there are natural killer cells. As the name indicates, natural killer cells naturally kill also any cell they find suspicious. For example, a lot of cancer cells, in their process to try to become a tumor, they precisely downregulate these MHC molecules. So now these naked cancer cells can no longer be effectively killed by T cells. So our bodies evolved a system to account for that. So when cells don't have MHC, they're going to get killed by NK cells. They need to have molecules to also inhibit the NK cells. And so you can see this becomes a pretty complicated puzzle very quickly because the immune system has so many cell types. When you get rid of one, you make the other one stronger. And so this is, very much work in progress. But what Sen has done is a kind of a proof of principle, which is three modifications. You take out B2M, that means no MHC class 1, we call it, to be recognized by CD8 killer T cells. They take out C2TA, that means no more MHC class 2 to be recognized by CD4 T helper cells. And they put in CD47, which is the don't eat me signal. So the macrophage is not going to touch these cells. Turns out that the natural killer cell is also partial to the CD47 molecule. So really, it's two birds with one stone. CD47 appears to inhibit, to block both macrophages and natural killer cells. So we have this, again, hypoimmune stem cell, and then its derivative. And so through a series of experiments and different models, so this is one that's going to be close to what I just told you about, which is you take cells from a human patient or in stem cells, and it can make humanized mice. That is, are this human type 1 diabetes model. And so what they show is that if you then look at the data, you see protection by being this hypoimmune type of cell. They've also used in monkeys. And so you see if you have these edited cells, you see the color code there. This is now in monkeys, in non-human primates. And so you see there, if you lose MHC class 1, the red goes away. And if you overexpress, really make a lot of CD47, the yellow goes up. And so you see this cell on the right, no red to be seen, and lots of yellow. And so these cells are the ones that if you put them in the monkeys, you see some stable level of C-peptide. Now, of course, these are in monkeys, and this is a cell number that is not curative, but it's sufficient to really show that this concept could work. And so, emboldened by this, they did the first trial in a human. This is a type 1 diabetes patient in Sweden. And so, what they did was they took cadaveric islets, cadaveric beta cells, so not stem cell derived yet, and they did these three gene edits that we talked about. They take out MHC or HLA in humans class 1, MHC or HLA class 2, and they put in ICD47. And so these three gene edits, they then take these beta cells and inject them in the arm of the type 1 diabetes patient. And so this is a small dose that they calculate is roughly 3% of what it would take to really cure this patient. So this patient still has to take insulin. What this patient does not take, though, is immunosuppressive drugs. If you recall from the beginning of our talk, tacrolimus, sirolimus, rapamycin, cyclosporin, none of that. None of those immunosuppressive toxic drugs. And so what they found is up to now 14 months, the cells are still there. And they make C-peptide, which is a marker for insulin. And so you have a small, but not zero, number of beta cells in a type 1 diabetes patient for over a year. And these cells make insulin. not enough insulin to cure the patient. We don't know how much longer they're going to be there for, but this is another piece of the puzzle, I think, and a system to show that these are the kinds of trials we need to be conducting to show safety and also some hints of, if not efficacy, that the mechanism works, right? So again, there's no efficacy here. The patient has to take just as much insulin as he had to before. But now he has his beta cells in his body that seem to be just fine in spite of the ongoing type 1 diabetes disease going on. And so it gives us tremendous help that all these gene editing in the stem cells and the beta cells and the regulatory T cells really is inching towards having a functional cure for type 1 diabetes that does not rely on immunosuppressive drugs and transplanting cadaveric islets. So that's really where the future and the present really is headed. So with that, thank you for your attention. I just have a quick slide. A lot of people to thank. Some of the work I showed is work from my own lab. So these are the people in my lab, Halloween, kayaking, with lab coats working sometimes too. Lots of collaborators. I'm at the Medical University of South Carolina, and so working with Dr. Doloff, for example, has been a lot of fun and productive. But also, as I mentioned, University of Florida, Dr. Holger Russ, T1D and stem cell and beta cell expert. At Clemson University, Dr. Ying Mei is an expert in regenerative medicine. Dr. Shaturji at UPenn with a protein language model, so using artificial intelligence to really take us to the next step in designing these lock-in key mechanisms. And then, of course, funding from Diabetes Research Connection, Breakthrough T1D, the NIDK, and others. And so with that, I thank you for your attention, and I see there's some questions in the chat, so happy to take any questions. Thank you.
Well, that was absolutely like a TED Talk for all of us. I'm going to put us into gallery mode, and I'm going to kick this over to Kelly to start some of the questions. This was so clear and understandable and engaging. Thank you. Kelly, you got it.
Awesome. Dr. Ferreira, thank you so much. That was amazing. I was able to tour Dr. Russ's labs years ago when he was still here in Colorado, and it's so exciting to see how much the work has progressed. So bravo. I'll say that we have on this call a whopping almost 1,400 years of lived experience with diabetes, which is really incredible. And I know, and it's really exciting to just see so much happen in the decades that we've all lived with this disease. I'll start with one question and then we'll go through maybe just a quick couple more. We might not be able to get to everyone. We did ask people to submit questions ahead of the talk, and one of the ones that came up over and over and over again with this audience in particular is, what's the likelihood for people who have been living with type 1 for a long time? Or, maybe is there an age cutoff for when these therapies might be available to our audience very specifically? Can you speak to that?
A little bit. So the goal is to start clinical trials before 2030. And it shouldn't really matter the stage because we bring in the beta cells and the regulatory T cells, right? And so it does not depend how many beta cells are left. There could be 0 beta cells left. We are bringing beta cells. Now, on the immune side, of course, as you might know, as our bodies age, so does our immune system. And so taking a regulatory T cell from someone at 80 versus 8 years of age might be different. And so that's what I think that another iteration of this technique is to make the whole thing come from stem cells. This way, all the cells are made in a laboratory. You don't have to worry about sourcing the cells. And even, one step further is that a lot of gene mutations in type 1 diabetes patients are in genes associated with regulatory T cell function. And so what if by taking regulatory T cells from a type 1 diabetes patient, those cells don't work as well as they should? Maybe instead you want to take Tregs, type 1 regulatory T cells from non-diabetic patients, but then they're not matched anymore, right? They get rejected. And so that's why I think that really implement this at scale to, as you asked yourself, to be for everybody, not just for a certain age, a certain stage, I think these have all have to be made from stem cells and be off the shelf. That's really the vision.
That's amazing. Thank you. And then Jamie and Jim, I think we're also going to pull some questions from the chat as well. And maybe we have time for about two more.
I think we'll extend it as long as we have some real good questions. So, and we'll
Yeah.
just keep going.
I'm just wondering if you can clarify, if these therapies take hold and they're working, is there a sense of how long they would stay working? Would it be a functional cure forever? Would there be a time period where some of these things might get turned off? Any thoughts about that?
Yeah, that's a great question. So I would say that there's a few unknowns. So one is the biology of the regulatory T cells. So in the textbook, there's two big properties that Tregs use to work. Forget about all the molecules. There's just two big commandments, if you will. One is called bystander suppression, meaning that any environment, microenvironment that the Treg is in, just by being there as a bystander, there is immune protection. Number two, more relevant here, is called infectious tolerance, meaning that tolerance can be propagated. So if you put a regulatory T cell in a specific environment, it can not only protect from immune rejection, but also convince other T cells to themselves become new regulatory T cells. It's like teaching other cells to become like itself. And so then the idea then is that after the cells that we inject have passed away, their effect lives on. Now, this has only been proven in mice. Humans live very long, so it's very hard to prove. And so because of this, I think an open question in science in general, at least in immunology, is do human Tregs observe this infectious tolerance property at scale? Because I don't think the cells that we put in are going to live forever. Maybe they don't even live a year. But if their effect keeps going, then you don't have to keep redosing, because you could see a scenario where you have your beta cells there, and beta cells are post-mitotic. They can last for a lifetime. But then the regulatory T cells, if they vanish, you'd have to inject new ones to keep protecting those beta cells. And it'll still be an improvement. Maybe you do the injection like every 6 months or every year instead of doing multiple injections a day. But this is a big question in the field, and so it's going to be hard to answer before clinical trials, because in mice, we went for a month, as you saw our own data. Now with these new humanized mice, we're hoping to go for 3 months, see if they last for 3 months. But even 3 months for a human, that's not that long. And so I think really you'll need clinical trials to answer your question, which is, how often are we going to have to be getting new Tregs and new beta cells? That's a great question. Yeah.
Great, thank you. Jim, I'll pass it over to you if you wanted to share a question, or I can jump in with another one.
Sure. I think there were a couple that were just around what is the hardest, most challenging portion of this therapy. Um, from all that you've shown, what's the biggest challenge?
That's a good question. The biggest challenge. I think a big challenge for sure will be to deploy at scale, specifically on the primary cell size, meaning the blood cell, because stem cells, as I mentioned, the field as a whole has become very proficient in making beta cells from stem cells. So you look at trials from Vertex, I didn't even mention what Vertex can make in Boston. They can make beta cells from stem cells that look almost like natural beta cells. And so we have that. Now, they don't have the protection part, so they use immunosuppressive drugs, they use encapsulation that doesn't work too well. And so, but the beta cell part, I think the field has pretty much figured that out. But now the immune component, this is what no one knows, to be honest, right? I mean, you have approaches that you make the beta cells hypoimmune, so you make them almost invisible to the immune system, but The immune system is very smart. It always catches up eventually. So how long can a cell stay invisible for before it gets destroyed? And a corollary of that is if you make a truly invisible cell to the immune system, if that cell ever becomes a cancer cell, you're in big, big trouble, right? And that's why those of us who are old-fashioned immunologists, we like to think about immune tolerance and immune education more so than just immune ignorance, if you will. And so we don't know yet how long hypoimmune beta cell can last. We don't know yet how long a Treg protected beta cell can last in a human. So, and the biggest challenge, I think, I hope I'm not repeating myself too much, but it's really that. The challenge will be how long can we obtain, effective protection of beta cells in a T1D patient? And this, I don't think we can really know before doing it, which may sound unsatisfying, but that is my current view on it. It's like without doing it, you don't really know.
Right. Thank you.
We probably have room for one or two more questions, so Kelly or Jamie.
Yeah, a question came in regarding whether any of the work you're talking about today would apply for newly diagnosed people who may still have existing beta cell function of their own cells.
That's a good question. So it could. So right now, if you look at, so if we forget for a second about the two-pronged approach of the bringing the beta cells, bringing the Tregs to protect the beta cells, there are trials in Poland, for example. I actually met the person in Tokyo last year where if you catch people very early and they still have a lot of beta cell mass left, and you give Tregs, those Tregs can protect the existing beta cells. Now, it seems to be more of a delay than really a cure. Similar to teplizumab, anti-CD3, same thing. You delay the disease by like 2 or 3 years, but people eventually, most of them anyway, still get it. And so there's definitely room there. Now, in our case, what you'd have to do is the approach that I mentioned is you put this bait, if you will, this flag in the stem cell, and then in the lab, this stem cell becomes a beta cell and you transplant it. You could also imagine instead having a way to inject in your body this flag and make it go only to your own beta cells. So then when you give this engineered Tregs, they protect your existing beta cells because they're going to find that ligand. And so this is semi-possible. It's a fast-moving field. There's a lot of efforts towards gene therapy. So gene therapy here understood as changing genes inside your body. Here I'm talking about cell therapy. I take cells from the body or from stem cells, I engineer them genetically and put them back in, so as cells. And then gene therapy is the gene itself is a therapy. And so there's a lot of effort right now, mostly in the cancer field, but I think it's going to permeate also to this, where can we deliver genes to cells inside your body? So in this case, you would deliver this unique flag to the beta cells inside the body and then no need to do stem cell-derived beta cells, right? Just put in the CAR Tregs intravenous, so that's easy, blood in, blood out, and then those would go and protect those existing beta cells. And so that would be the way to do it. But I don't want to claim that I personally have not yet modified beta cells inside the body, only outside of the body. But there are people, there are groups working on this. We have different virus serotypes, right, that are kind of semi-specific. So if you inject them in someone's body, they preferentially introduce themselves to beta cells. And so you could imagine that being something. Yeah.
Thank you.
I think this has been beyond extraordinary. The comments are very telling. I'm old enough to remember the movie The Incredible Journey with Raquel Welch when they were shrunken down into a little spaceship that went into your veins. And this feels like we're living it. So thank you for that. Thank you for making it so very clear. it was a perfect for our audience. And so my question is, maybe the final question is, what can we do in the community? I feel it's real important for us to support and encourage and be available to the research community, because you are the ones that make things happen, that make our lives better. So if there is anything we should be knowing or doing, please let us know. We can rally people. We can do stuff.
Thank you. No, I think the T1D community is extremely active and really a pleasure to interact with people in San Diego, in Boston, in Charleston. There's a South Carolina chapter for Breakthrough T1D here in South Carolina. And so it's really, as you said, a lot can be done. And so there's a few things. I mean, I think the big thing is that science is a rather slow process. And so it really needs that sort of continuous, congruent funding to just, keep going and going, because no one knows where the next big thing is going to come from. I mean, you might have heard of CRISPR, for example. CRISPR now, the first therapy for sickle cell disease, use CRISPR, right? CRISPR came from yogurt because they don't want to lose bacteria to make yogurt. And so bacteria use this tiny defense system called CRISPR to kill viruses. And so from that, someone took the jump into making a tool that can change human genes. And here, for example, right, I have a lot of research on stem cells and mice and weird diseases and all this. And so because of that, I think that the big thing really is to just, I think the United States in general is a very special place to do science because there's so many avenues of support, right? We have foundations, we have interest groups, we have the federal government, different organizations. And so I would say that's probably the biggest thing that people can do, I think, is continue to advocate for, we need more funding, also more education. People who don't know what's going on, they don't really appreciate what can be done, what can be done yet. And I think that's also sometimes a gap. And I think part of this is probably also the scientist's fault, but one cannot do everything at once. But so I think those are two big things. I think one is always funding to keep that consistent stream of ideas and tests, right? You always need to have a new idea, test it, failed, have another one because that failed, now works a little bit. And it takes months and years to come up with something good. And then secondly, yeah, just educating people for what the disease is and what stage are different therapies. I think again, of course, I'm a bit biased, but I think the field as a whole is really coming together. Many pieces of the puzzle are coming together to have a really good therapies, right? To have Vertex and others making stem cell-derived beta cells, we have Sana making hypoimmune beta cells. We ourselves work with regulatory T cells to protect them. There's also some work on encapsulation, right? Kind of having some sort of protective layer, if you will, to the beta cells. And so it's really always inspiring to go to the American Diabetes Association meeting every year because there's always, it feels like, quite some progress in the past few years that I've been attending and speaking at it. And so, yeah, so I think we're on the brink of something. Now, of course, I'm not a physician or a fortune teller, so I can't tell you it's going to be next year or it's going to be the after. So this I don't know, right? But at least the science has become increasingly exciting. And so I think the big thing again is to make sure it doesn't stop now, right? Because if it
We
stops,
hear you. We hear you 1,000%. The comment is we can always advocate and we can support science. And this recording will be published on our YouTube channel and shared. You made it so clear that we actually understand what you're all talking about, which has been so helpful. One real quick question, because I think it's an interesting question, is does the Islet Act matter to you?
whether to classify islets as cells or as drugs. Yeah, I think it's a very interesting point. So, also coming from the CAR T cell world, where CAR T cells are drugs, and some people argue that makes them that much more expensive. Right, a CAR T cell dose for leukemia is $500,000. That's how much it was in 2017. That's how much it is today. But in my mind, technology should get cheaper over time, not flat. right? And I think likewise, islets, of course, islets are cells. And so this could help to do that because bone marrow transplantation, which is by definition the first stem cell therapy there is, it's because it's the blood stem cell, right? That's been going for many decades, never FDA approved. It's a highly regulated experimental therapy that uses cells. And so because cells are regulated differently from drugs, there could be quite a gigantic difference in costs to patients and to practitioners, whether you call something a drug or a cell. And so, not being a physician or an insurance reimbursement payer, that's where I see some very, potentially big impact there. Just the cost of goods.
So excellent. I think I'll be buzzing all night. Thank you. Thank you so very much for your time. Thank you for teaching us tonight. Thank you everyone for joining, and we'll be back to you, Dr. Ferreira. Thank you. Good night,
Thank you
everybody.
for the opportunity.